Assalamualaikum warahmatullahi wabarakatuh. Dear students, I am Tanveer Iqbal, teacher by High School Shatigam. Class for 8th. Today we'll start our new chapter, the cell. As we all know, cells are the basic structural units of an organism. Whether it is a multicellular organism, whether it is single-celled organism, any organism in this universe, in this world, is made up of tiny small things. These are called cells. Just as buildings are made up of bricks or buildings are made up of stones, they make up and then make a fine structure, and that structure is useful for us. Same ways the cells mix up with one another and function properly so that this organism lives very well and fits in this universe. So all living things are made up of tiny living parts called cells. We cannot see those cells from the external examination of a living being. If we see the external parts, we can see only different parts. If we take the case of plant, we can see the root, we can see the stem, we can see the leaves, we can see the flower, the parts of the plant. If we take the case of humans, we can see head, face, arms, legs, chest, abdomen, etc. So then how do we see these cells? We see these cells by studying the internal structure of different parts of the human being. We can make thin fine sections of the various parts which we want to study, and then we can observe these with the help of instruments like magnifying glass, like microscope, and microscope has many types. So first of all, we can see the instruments by which we can use to magnify these objects so that we can see the cells. First of all is the hand lens. Hand lens होता है glass. It is a glass. It is convex glass on both sides. Convex glass on both sides. You will study these in the light. So it's a biconvex lens. आज पहले ज्योमेट्री आते थे, ज्योमेट्री में आते थे, उसको बोलते थे हम कश्मीरी में आतशीश. वही आतशीश जो होता है, ये होता है बाइकन्वेक्स लेंस, ये होता है मैग्नीफाइंग गिलास. अगर हम ये रखेंगे कि सन की रेज में ये सन की रियल इमेज बनाता है नीचे जहाँ पे रेज पड़ती हैं. अगर पेपर नीचे होगा या ड्राई लीव्स होंगे, जब रियल इमेज बनती है सन की तो बराबर वो जल जाएंगे. इट इज़ अ फ्रेम एंड इट इज़ अ हैंडल. तो मैंने बनाई भी है. ऐसे होता है वो. ज्योमेट्री में आता था पहले, आतशीश बोलते थे, मैग्नीफाइंग गिलास होता था वो. दूसरा है, सबको पता है, माइक्रोस्कोप. माइक्रोस्कोप जब ईजाद हुआ उसके बाद अच्छी तरह से दिखने लगी कि सेल कैसी होती हैं. तो माइक्रोस्कोप हमारी लेबॉर्ट्री में भी है, आपने ऑलरेडी भी देखा होगा कहीं. तो उसमें बहुत सारे पार्ट्स होते हैं. आई पीस होता है, ड्रा ट्यूब होता है, बॉडी ट्यूब होता है, ऑब्जेक्टिव्स होता है, स्टेज होता है, मिरर होता है, एडजस्टमेंट्स होते हैं, कोर्स एडजस्टमेंट, फाइन एडजस्टमेंट. तो हम आराम से देखते हैं. जो स्लाइड होती है उस पर हम वो चीज़ रखेंगे जो हमें देखना है. ओनियन पील का देखना है तो ओनियन पील का बहुत सी थिन लेयर उठाएंगे. लीव्स का देखना है तो लीव्स की बहुत थिन लेयर उठाएंगे. हमारे अंदर का जो ये स्किन लेयर होती है ये देखने है तो फाइन लेयर उठानी है फोरसेप्स से, तो डालनी है स्लाइड पे. फिर हम कोई ड्रॉप भी डाल सकते हैं आयोडीन जिससे वो अच्छी तरह दिख जाएगा. तो when we will inshallah go to school, we will definitely see the cell structure under the microscope, which is already present in our laboratory. We are also to see the function, how that microscope functions, how we can make the coarse adjustment and how we can make the fine adjustment, how to make the slides and how to fit the slides on that stage. It has two clips. So how to fit that slide so that we can see the clearer image of the object which we want to see, which we want to observe. So when we can see under the hand lens and we can see them under the microscope, we can see the cell structure easily. What the cell already consists of, or what the organism is made up of the cell, and the cell is in turn made up of many organelles. These organelles can also be studied under the microscope if that is a good microscope, like there are mitochondria, like plastids, etc. Now, taking the case of the organisms that are made up of cells. Organisms can be one-celled, single-celled. So bacteria, amoeba, if we take the case of animals, if we take the case of plants, Spirogyra, etc. These are unicellular organisms, and all the functions are carried by the single cell. All types of functions are carried by the single cell. When we talk of multicellular organisms, there are billions and billions of cells working together. They make the best case of division of labor if we take the case of cells. So plants and animals consist of billions and trillions of cells. These are multicellular organisms. These cells, when mixed up and do the same function, they make tissues, then they make organs, then they make organ systems, and then they make organism. So we have to study a cell is the basic unit of living things. Cells in living organisms have often been compared with bricks in a building. जैसे हमारी बिल्डिंग में शेर से ना शेर आसान साई बिल्डिंग मिल शेर शेर आना पैठ तिल से बना बिल्डिंग अपनी ही स्कूल की तरफ देखो. When these bricks are stacked together uniformly, they make a good building, and that building uses the purpose for which that is made for. So just as bricks in a building are the basic structural units, cells are the basic structural units in living organisms. Further, just as buildings are different from one another, organisms are also different from one another. Cells have been in existence since origin of life, which took place millions of years ago. Majority of cells are too small in size, which cannot be seen with the unaided eye. If we want to see the big cell which we can see with our unaided eye, there is the example of an egg. An egg is a cell which we can see with unaided eye. That is a single cell, or that is the structure of the cell. We can see the objects as small as 1/1000th of a millimeter can be seen with the help of a microscope. Now, taking the case of discovery of the cell discovery, Robert Hooke in 1665 discovered the cell. He was an English scientist. What did he observe? He observed thin slices of cork, which are the box-like compartments forming a honeycomb structure under the cube microscope. His microscope, that was not the good one, but still he saw the things. He named these compartments of cells. Yesterday or day before yesterday, we were studying apiculture. When the honeybees make honeycombs, they make that structure. Same ways, the cork was made up of box-like compartments forming a honeycomb structure. He demonstrated his work to scientists at Royal Science Academy of London. I have made that what did he observe under there. I have tried it. If you want to see on the textbook, here it is called as observed by Robert Hooke. Okay.